right, welcome everybody. So let's talk a little bit about chemistry. This is one of my favoriteest things to talk about. So I'm really excited to be here and share a little bit of this. So the topic for the talk is chemical characterization: how to prepare for the future in this time of transition. And it really is a transition time for chemistry in medical devices. The FDA guidance on the application of ISO 10993 really hasn't been out for that long. And when it came out now, I guess 2 years ago or so, I remember reading it with my highlighter to highlight everything that talked about chemistry with medical devices in it. And after just a little while, I had to just throw the highlighter away because chemistry is just all through this document. So there's a really strong motivation for for doing chemistry with medical devices. It's highly recommended, but the guidance on what exactly to do or the details about what you should do have been a little bit vague, to say the least. So there is an ISO document that talks about how to do chemical characterization. This is ISO 10993-18. This document has it's under draft review now, so I actually have a copy of this new draft on my desk to look at. And we think that we're very close to having it finished. But the current version that's out is also pretty vague. So there's actually a whole lot of different things that you can do when it comes to chemical characterization for medical devices. And since there's been an increased focus on this, things really have been shifting over the last couple of years, and we find ourselves right now in an environment where the expectations of regulators and what people plan to do is shifting very rapidly. So that's the focus of this talk. Before I go too far, let me first just give a personal introduction. So basically, chemistry has been my whole life. I've just been a super nerd right from the beginning. I was more interested in playing with my chemistry set in my basement while all the other kids were getting sunshine and stuff. And basically, I just can't leave it alone. My background is more academic, so I got a PhD in physical chemistry, and then went and taught chemistry for a while before going to Europe. I did a lot of research in micro and nano structures that have cool functionality with light before heading to Nielsen Labs, and really devoting my my expertise towards helping medical devices be better. But my one claim to fame is that I once was in a Mountain Dew commercial. But that's me holding a bottle of Mountain Dew Whiteout, probably the second best flavor of Mountain Dew. And because of my love for Mountain Dew, people tend to call me Dr. Dew. So I recommend, you're free to call me Dr. Dew if you want. I will answer to either Matt or Dr. Dew, and it's no problem. So about chemistry for medical devices. So when we talk about chemistry for medical devices in the context of medical devices, it's really important to remember that this is part of a bigger picture of biocompatibility for a medical device overall. And biocompatibility it's a big topic that involves lots of different pieces, and chemistry is one part of that. So I think that this picture of the consulting team here at Nielsen Labs really kind of highlights how this is a bigger thing. So that's me in the middle there. So I focus mainly on chemistry for medical devices. The person on the far left there, that's Thor Rollins. He's maybe the godfather of biocomp for medical devices, and not just because of his age. It's really more because he's really had his hands in all of these different standards that come into play when it comes to biocomp. So truly an expert. The person with him, Audrey, she's another biocomp expert, and the two on the right there are toxicologists. So it's important to understand extractable leachables chemistry in this context with toxicology in mind and biocompatibility. So these subjects are way too large to talk about in just 45 minutes. Even just the chemistry, trust me, I could just go on and on and on and on. But that's why we have other seminars coming as well. So Thor Rollins, as I said, truly an expert in biocomp, will be speaking tomorrow here at MDM West. So he's there. We also have a very rigorous seminar schedule coming up. So we have a biocomp seminar coming in Las Vegas. So that's coming up. It's in the lineup. and other biocomp seminars as well. So in case you're interested in biocompatibility, which we all should be because this is a big part of it, those are resources that are available for you as well. Okay, so here's an outline of the talk. This is what we want to focus on when it comes to E&L. Always keeping in mind that this is a piece of a bigger picture here. So first of all, we'll talk about extractable, leachable testing strategies across the spectrum. So as I said, the guidance available is a little bit ambiguous, so there's a variety of things that can be done. We'll talk about the evaluation of chemistry results for regulatory approval, and then we'll talk about the approach that we've taken so far at Nelson Labs in terms of trying to balance the need for scientific quality and adequate biological safety evaluation with the needs of the manufacturer and meeting regulatory requirements. So I'll talk about changes that we're seeing throughout the conversation, but then at the very end I'll re-summarize these. Okay, so before I go any further, I really should define what E&L is, because I'm going to use this term like 1,000 times during the presentation. So it's really important that we understand what we're talking about here. So extractables and leachables. Extractables are compounds that can come off of the device or leave a device when the conditions are exaggerated compared to clinical use. So if we expose the device to an aggressive solvent at an elevated temperature that's much worse than what the patient would actually see, that would be an extractable. Leachables, on the other hand, are compounds that we would really expect to come off during clinical use. These are compounds, if we tried to simulate what blood is like at the temperature of blood, and we would measure it that way. I also just want to briefly note, you don't need to take too many notes during this talk. If you want, you can give me your business card after the presentation. I'd be happy to send these slides to you, so you don't need to worry about writing down everything that I'm saying here. Yeah, so those are extractables and leachables. That's what we're talking about. The reason why we do this is because, as I think everybody in the room probably knows, before you can market a medical device, we need to demonstrate that it's biocompatible. And this biocompatibility needs to be demonstrated per ISO 10993. And as a manufacturer, you have a range of options available to try to demonstrate this biocompatibility. So, one route involves primarily going down this list in the ISO 10993 and kind of checkboxing all of those biological endpoints. So that's an option that's available. Another option is to really look at the device materials and its history of use and try to make some sort of justification to the FDA or a regulatory body doing as little testing as possible. Chemistry can be used and really should be used as part of this assessment along with some of the other things. So there's a variety of options available to approach biocomp. Usually chemistry is one of these options that's part of this evaluation that should be considered, right? And I guess the important thing to remember is that we're doing chemistry in support of biocompatibility, so we're trying to address specific biological endpoints that might need to be addressed. It's not necessarily from a hardcore analytical perspective, knowing exactly everything that's in the device or the material. So we only care about the relevant things. Okay, so as I mentioned before, the guidance so far has been pretty vague. There's some flexibility about what we can do. And when I start to think about the different strategies available when it comes to chemistry, I like to think about it in terms of these techniques' predictive quality, the scientific quality of what we're doing, versus how burdensome what we're doing is. And to put this in the context of biocompatibility, you can think of animal studies being on this graph somewhere here. So animal studies remain a valid option to assess biocompatibility. They are relatively burdensome compared to a lot of the chemistry that we're talking about doing. And the scientific quality, while there is some value there, it's a little bit on the lower side. So the reason why this figure is important is that this kind of represents a burden barrier, if you want, to manufacturers. So long as we have a choice between doing chemistry and animal studies, if the chemistry that we're doing, even though it's awesome, if it becomes more burdensome than just doing the animal tests, it starts to feel like a good business decision to save time and money and just do the animal tests rather than the chemistry. So that's something to keep in mind. If I were to plot chemistry strategies on this graph, I think it would look something like this. So on the low end, you can see there's options there that are very inexpensive and fast, but they don't, provide a lot of, scientific quality or predictability. And then, I think that there really are lots of options that provide better information or better data than the animal studies, but are still quite a lot less burdensome. Then, of course, chemistry is so awesome that you can always add another task and do it like a more detailed analysis, and eventually it can get up there where it's more burdensome than doing the animal test. So there's kind of an evolution here in these strategies. If I think about the demand, then I would say, what we see is that manufacturers really want to do better scientific work than what you get from an animal study. I think it's clear that, when we do a test in a rabbit, but the device is for, say, a neonate, that, these aren't like baby rabbits and the results don't really, correlate, or, the sensitivity for a genotox test might not be as good as chemistry and so on. So I think that there's demand to do better scientific work, and people are, we can tolerate a little bit of, increased cost to do better work, but as soon as the testing burden exceeds the burden of animal testing, then it just becomes a bad business decision to choose it. So where we target, we want to be here, we want to do the best scientific quality that we can while still being less burdensome than doing the animal test. And this is in contrast to, say, like some pharma approaches, which can be, very, very extensive, and maybe more burdensome from a testing perspective than animal studies, but this is the level that's required for those types of studies. So along this line, we can think about different analytical strategies and different tasks that could be included. I summarize these here, so you can imagine on the very, very low end, things that are cheap and fast but not very predictive are things like gravimetric NVR. So gravimetric NVR is a method that doesn't have great sensitivity. Whatever you find there, you don't know what it is. So it's kind of difficult for a toxicologist to do much with that data. A step up from that would be something like FTIR or TOC, THC, the total hydrocarbon, or UV-Vis. This is getting a little bit better, but when you have complicated mixtures of compounds, things get to be a little bit tricky. Going further up, you can start to talk about semi-quantitative mass spec methods. So anytime you start to combine chromatography and mass spectrometry, then we're getting a little bit closer to something that's really useful to a toxicologist that might be able to, might be wanting this data. And then, finally, you add more and more layers to this. So you can have multiple mass spec methods with at least some fully quantitative results. And then there at the end, you can go more like a pharma route where the goal would be to fully identify and fully quantify every single compound that could come from the device. So the strategy that that we take generally is this one where we have multiple quantitative methods, but we don't need to go so far as to try to nail down every single compound that might come from the device, because we'll balance sort of like the risk versus the benefit of doing that. Okay, so just to to keep this in context, so the goal is to support device safety at relevant biological endpoints using analytical chemistry testing. In the process, if we do it right, we spare animal life, we save cost, time, and money. So I think in all of those, we can keep all of those things true. If we're looking at doing chemistry more from like an engineering perspective, then once you do this, you really gain a lot of understanding about your device materials and processing and things like that. So we find a lot of use in those situations as well. Okay, so I think it's best to look at this with an example. So I mean, we'll take a pretty serious example here. Uh, take take this Spock hand, right? So the Spock hand is a medical device. Its intended application is to be implanted into the body next to the heart so that you can keep the memory of Leonard Nimoy with you always, right? Now, if you were going to be the recipient of this device, what type of testing or assurance for its safety would you want, right? So if we're thinking about chemistry in this case, would you say, well, I know that heavy metals can be bad, so let's just like screen against lead? Or would you say, well, it's plastic, maybe there's DHP in there, let's just screen against DHP? No, right? If you're going to get a life, altering implant, something that's going to be with you all the time, you would be concerned or wondering about anything that could leave the device. Even stuff that you don't know that's in the device. You'd be like, well, what about, I know that there's DHP, but what about the stuff I don't know? And so this really defines the scope of the work, right? So we need to do chemistry in a way where we cast a very, very wide net, where we screen for as many different things as possible, even the things that we don't know that might be in there. And we need to do it in a way that still is more cost effective than doing animal tests. So that's the idea. We look for any substance that could leave the device and enter the body. This goal really defines the analytical approach. So you can imagine we start to think about anything that could leave the device. Some things are going to be soluble in water. Some things are only going to be soluble in a non-polar solvent like hexane. So we use both solvents. We'll extract in water and hexane, oftentimes even a third solvent like an alcohol, and that way we'll be sure that whatever comes out will at least be soluble in the liquid that we're using. And then we'll take these liquids and we'll analyze them using a variety of different methods. Another thing that we're going to do is when we start to extract them, we'll do this so-called exhaustive extraction. So we'll try to make sure that we're getting as much of these compounds out of the device as we can. So the way that this works, and this is a point that we're seeing the FDA ask for more and more. So we didn't always used to do an exhaustive extraction for these devices. Now we're seeing the FDA want this more and more, and even just the last weeks we've started to roll this out all the time when we have permanently implanted devices. So an exhaustive extraction works by taking the same device and extracting it over and over again in fresh solvent and analyzing those solvents and seeing when the subsequent extraction is less than 10% of the initial one. What this allows us to do is it allows us to trend how compounds are leaving the device. We can make sure this is going down, approaching a level that's stable. If it's going up, then there's a problem, right? That means that the device is like degrading and it's not stable. We should really take a step back and catch it there. So we'll do an exhaustive extraction in all three solvents, water, and alcohol, and hexane. And then we'll take those and we'll analyze them using a variety of different means. So, in chemistry, we really love to group compounds together, and there's many different ways to do that. I of course, love this stuff. When we're thinking about analytical methods, it can be useful to group these compounds by, first of all, whether they're organic or inorganic. So organic is, of course, anything that contains, carbon or is carbon-based. The inorganic ones are the opposite, not carbon-based, so they all have metals. For the inorganic side, it's pretty relatively easy to make an analytical method. We can do a method that just looks for metals. This is inductively coupled plasma mass spectrometry. Basically, we take the extract, we burn it at temperatures hotter than the sun, so it's just metal ions. Then we can sort those by mass. All these metals have different and very precisely known masses, so we can identify what metals are there that way and then find out how much. So that takes care of a whole category. On the organic side, at least for analytical methods, it's useful to group things together by how easily they evaporate. So for very small and light easily vaporizable molecules. We have a method, it's gas chromatography with mass spectrometry, GCMS, headspace. This is where we have a vial of a liquid and we sample the air above it, and anything that's volatile enough to leave the liquid we measure. For medium-sized molecules, this is actually the broadest single analysis that we can do. We have a direct injection method. So it's, again, GCMS, but it's injected directly into the machine. Using this one method, we can screen for approximately 300,000 different types of molecules. So it's really broad. Once we get much bigger than medium-sized molecules, and medium means, say, so it would include DHP, but too much farther than that. If they start to be like really heavy, sticky molecules, then GCMS just isn't a good option, because once it gets into the machine, it can't really vaporize, and it just stays in there. You don't see it. So for those, we would use LCMS. So there, the molecules stay dissolved in a liquid, the liquid gets carried through the machine and are separated that way. LCMS is kind of tricky because, for GCMS, there's these big libraries that we can use that allow us to identify what comes off of the machine. For LCMS, we can't really use those big libraries. You have to have physically run the compound on your own machine in order to identify it. So you really have to have a good database on hand or available to do LCMS and have it be very effective. If you have a good database, then it's a great tool to look at what larger molecules could be coming from your device. Once you go through this process, analyzing the extracts using all of these different methods, then you're left with these results, right? So this is the end point for chemical characterization, is a report that lists, say, all the different compounds that came from the device and at how much. And then here, things kind of like diverge, where it kind of depends what you want to do with the results, which direction that you'll go. So if you're doing this as kind of an engineering project where you want to understand, say you want to claim that there's no DHP in your device and your supplier said there's no DHP, you want to kind of like check if that's true, then this might be a stopping point. Or if you're trying to control something that you're cleaning for, this could be a stopping point as well. But more often than not, if you're doing chemistry to support biocompatibility, then you need to go a few steps further. So that's where these results should be analyzed and assessed and understood by somebody who's qualified to make, say, some toxicological conclusions about what the numbers mean. So for that, you may have an in-house toxicologist that can understand that, or you can go through a consulting firm. So we have our toxicologists in-house at Nielsen Labs that are very adept at doing this. And they take those results and they say, okay, based off of these results and the intended use of the device, we can conclude it's not genotoxic, or it's not going to cause subacute, subchronic toxicity, and so on. These bullet points that the FDA really wants to see being addressed. Yeah, and so one of the things that I try to focus on is, as you go through to design a strategy, if you're in this situation where you're ready to perform biocompatibility for your device, and that you want to use chemistry because it's going to save you time and money and spare animals and all of that, then you need to somehow pick a strategy, and you're going to talk to probably a few different people on like what strategies they would recommend. And this can be kind of tough to pick chemistry, or to pick the right chemistry to meet your goal. So as you navigate this landscape, what I would suggest is, first, you should start by having a conversation with your toxicologist. So it really should start there, because if you have a device with special toxicological considerations, the toxicologist might say something like, well, I can't make a claim about genotoxicity unless this analysis is at least this sensitive. Or they might say, well, this device is a special risk for whatever. We need to make sure that we do a phthalates screen on it. And it's really important to have that information up front, because it's kind of frustrating if you go through all this chemistry and you do it away at the end, the toxicologist says, well, it's not sensitive enough, I can't use it, right? So start by having a conversation with your toxicologist. That should be the beginning point. As you go through, you need to think about the extraction solvents, what solvents are going to be used, how many solvents should you use. Historically, we've typically done just 2 solvents at Nielsen Labs. Now we're doing 3 because we see this being requested. You should ask yourself, do I need to do exhaustive extraction as part of this? Exhaustive extraction can be useful, but it also adds kind of a lot of time and some extra cost to the whole program. So that's something that the toxicologist can help you decide as well. And then when you start to consider analytical methods, you really should ask yourself about the quantitative nature of the results. And you should ask your lab about the quantitative nature of the results, because this can make a big difference, right? So not every result is reported fully quantitatively. It's kind of hard to make these results quantitative because a method has to go through this big validation process and so on. So you need to know up front, okay, do I need fully quantitative numbers, or am I okay with estimates, or am I okay with, if it's not quantitative at all, just like a true or false, this compound is present versus not present. And then you have to ask if LCMS is really necessary for the project that you're working on. So if you're talking to a lab and they're going to propose, say, an LCMS method, but they only screen against 10 compounds, and it's pretty unlikely that those 10 compounds are going to come up as a hit, it might not be scientifically justified to go through that expense. So let's just kind of walk through this with an example, right? So this is something that comes up for us from time to time. This is an enteral feeding tube. It's used to provide nourishment to underweight babies. Based on the cumulative exposure, while the tube can be exchanged every 7 days, it definitely can be used for more than 30 days cumulatively, so it's a permanent exposure device. So when this one hit our desks, first of all, we started with a conversation with the toxicologist. So our toxicologist, Dr. Campbell, she has a lot of history with research with neonates, so she's especially sensitive about these populations. And she said, okay, well, if this is the case, I'm going to want the sensitivity that much lower because the weight of these babies is that much lower, and she's going to consider that in her assessment. So we knew at the beginning, okay, we're not going to use our typical sensitivities. We're going to use one low enough that it can account for any possible adverse effects in a neonate. And then when we look at the extraction solvents, since it's a plastic device, it's a sensitive population. We know that the FDA is going to really be looking at it closely. We would recommend 3 solvents. So we do water. This formula is mostly water, but there's also a lot of fatty fats and stuff inside. So we do water. We do a midpolar and a nonpolar solvent. So we should ask, is exhaustive extraction really necessary? In this case, based off of the way that the toxicologist might assess the results, we might say, okay, well, from a scientific standpoint, we feel okay without it, but knowing that the FDA is becoming more and more sensitive to this, at this point I would recommend that you do the exhaustive extraction determination on that as well. And then when it comes to analytical methods, do we need fully quantitative results? In this case, the toxicologist was like, no, I'm not okay with an estimate on this one, because if there's 5 micrograms of this compound or 8, it could make or break the deal for me about whether or not I can sign my name to it and say that it's safe. So we went, fully quantitative. We recommend LCMS, and that would be the strategy we would use for something that's for a more sensitive population, a more high-risk population. Definitely when the device is permanently contacting, this is the way to go. Okay, for something else, say something like this is a wound dressing. So wound dressings can be used, I guess we'll call it prolonged, right? So this is really used to pack in a cavity wound to prevent it from closing and causing an abscess. So there is some risk there. you should still consult with the toxicologist, see what her sensitivity is. Since the device is prolonged, a lot of these acceptable limits or toxicological thresholds, they might be quite a lot higher. And actually, the sensitivity for this analysis could be less rigorous than what you would do for a permanently contacting device for a neonate. And what that means for you is that actually when the sensitivity is higher, right, and that means less compounds need to be analyzed and reported, it actually can get a little less expensive and easier to do. So, the sensitivity comes into play there. For exhaustive extraction, right now, we're not really seeing a lot of requests for exhaustive extraction from the FDA unless it's a permanently contacting device. So I don't think we would do it in this case. Probably just use 2 solvents. We'd run the range from water and then do the second one at hexane. And then when it comes to analytical methods, where the thresholds might be so high, it might not be such a big deal if some of the compounds reported are estimates. Maybe we can live with that in this case. And for LCMS, if it's very burdensome to do, adds a lot of time and cost, then we really should maybe balance the risk. It's a low risk device. The limits are high. We're doing other testing to complement the chemistry, of course. Maybe it's not so necessary in this case. So the strategy that you choose, I guess the takeaway here is that it can vary based off of the patient population and the contact types. You definitely should know whoever you use for a toxicologist, you should know what they want, because in the end they need to sign their name to this to say, okay, yes, I agree this is safe, or no, I don't think it is safe. They really kind of hold a lot of cards there. And then always remember that the way that this will work overall is that you'll do your chemistry, it'll be assessed by a toxicologist, their report will go with other reports, the results of these in vitro tests, or if you do have to do some of the other animal tests, all of those reports go together into a final big summary report, a biological evaluation report, and that's what actually goes to the FDA. So I mentioned some of the things as I was going along that are changing, that we see changing now. One of the things really important to note, this ISO 10993-18 is under revision. There's a lot of things in there that are changing. I see a lot of what I consider improvements to the draft. We hope to have this out soon, though I guess we've been hoping that for a little while, but I think we're close. We really focus on a risk-based approach. This concept of an analytical evaluation threshold is really prominent in this draft. It means that we really only need to care about looking for things that might have a toxicological consequence. I think that makes a lot of sense. We've been seeing lately a lot of requests from the FDA for exhaustive extraction for any permanently contacting device. So this is one of these areas where these changes have been coming a little bit more recently and pretty rapidly. So it's something that we recommend. We also see an increased desire by regulators to use 3 solvents. So in the past, we felt pretty good about spanning this range from polar, very polar with water, to very non-polar with hexane, and saying, okay, well, anything will land in between. Now we're seeing a lot more requests to include the mid-polar, even if you include a non-polar solvent. So this is something that's coming up that you should be aware of. Of course, the exception there is that if you damage or degrade the device, clearly that solvent isn't going to be relevant from a toxicological perspective. So if it degrades the device, that can be omitted. And then we also see an increased desire by regulators to see this LCMS included as one of the analytical methods for the device. They really want to make sure that those non-volatiles are being hit. Okay, so with that, I think I'll close, and I'd be happy to answer any questions that you might have. Yeah, please. Yeah. It's a little bit quiet, but. Yeah, yeah, so I mean I agree completely with everything that was just said. So I think that one of the greatest strengths for extractable leachable chemistry, one of its strongest motivations, is the fact that we know pretty convincingly that a lot of these animal tests really aren't sensitive enough to predict what we want them to predict. So we're really increasing patient safety by including these methods. I think it's a very strong motivation for this. Yeah. Yeah, I agree. I think we're on a very very good path here, and I think that we ought to continue, going down this path. Obviously, that's the direction that we're going. So yeah, thank you. Yeah, any other questions before we break up? Yeah. I mean that's a great question, and actually I've been getting a lot of requests for packaging testing lately as well. So when it comes to biocompatibility and extractable leachables chemistry to support the safety of a device, then we always want to test on the final finished form of the device, final finished package, sterilized, the whole thing. Where this packaging starts to come into play is a couple of places. People might want to test their packaging ahead of that to just screen it to make sure that they're not going to run into a biocomp problem at the very end due to their packaging. And another thing that comes up that I see is that people will go through the entire gamut of biocomp testing for their device and pass everything, and then want to change their packaging. And there, in those cases, they might do something like a packaging comparison test, where they'll test both packages and make sure that there isn't anything new and troublesome coming from the new packaging. But yeah, it's a great comment. Okay, any other questions or thoughts? Like I say, Thor and I will be here tomorrow. We're giving a seminar and a hands-on workshop tomorrow during lunchtime, and so I'd be happy to talk with any of you one-on-one if you have any questions as well.